Hi students, today in this class we will learn about degeneration and regeneration of nerve fibers. Our learning objective will be classification of nerve injuries and injury to the cell body, injury to the nerve cell processes, and the Wallerian degeneration and regeneration. See this? This is one axon with myelin sheath. It is enveloped by or covered by endoneurium. Some axons will form one fascicle. The entire fascicle will be covered by perineurium. And some fascicles together, they are covered by epineurium. This entire thing forms one nerve. See the coverings of the nerve. The whole nerve is covered by a tubular sheath which is formed by areolar membrane, that is called epineurium. Each fascicle is covered by perineurium and each nerve fiber is covered by endoneurium. So how myelination is formed in the peripheral nervous system and central nervous system? The central nervous system, oligodendrocytes will form the myelin sheath, and in the peripheral nervous system, Schwann cells will form the myelin sheaths. So many Schwann cells are involved in formation of myelin sheath for the one axon, whereas here one oligodendrocyte will give myelin sheath to many nerve fibers. The same picture is given here. PNS is having Schwann cells which are giving myelin sheath. In the CNS we have oligodendrocytes which are forming myelin sheath. So when the axon of the neuron is injured, what happens? Series of degenerative changes are seen at 3 levels. What are those 3 levels? One level is the axon which is distal to the injury, and one is the axon proximal to the injury, and another one is the changes in the cell body. Okay, so axon which is distal to the injury will show some changes, axon proximal to the injury will show some changes, and cell body will show some changes. All these are degenerative changes. So along with these degenerative changes, regeneration will also starts. The effects of injury to a nerve and the occurrence of regeneration thereafter, it will depend upon the degree and type of the damage. What are the common causes of nerve injury? First, it may be a transection, or it may be crushing of the nerve fibers, or local injection of toxic substances, or maybe ischemia due to obstruction of blood flow, and hyperpyrexia on the neurons, effects of increased temperature on the neurons. Then we came to grading of the nerve injury. There are two classifications of grading of nerve injury. One is the Seddon classification and second is the Sunderland classification. So according to Seddon classification, which was given in the 1942, the grading of nerve injury is divided into 3 types: neuropraxia, axonotmesis and neurotmesis. What is neuropraxia? See here, this is endoneurium, this is perineurium and this is epineurium. What happens in neuropraxia? It is having a conduction block, just a conduction block. So only the nerve conduction is blocked. It is a simplest or mildest form of nerve injury, okay, in which you may experience some motor loss and sensory loss depending on the type of nerve, and this is reversible and it has physiological conduction block. So physiologically there is conduction block. Why it occurs? It occurs mostly due to mechanical pressure, okay, mechanical pressure, and spontaneous recovery is possible after few days or few weeks. So for example, Saturday night palsy, okay, and tourniquet palsy, you can see neuropraxia due to mechanical pressure exerted on the nerve. Next, axonotmesis. What is meant by this? It is more severe than the neuropraxia, and it is usually occurs in the crush injuries. There is complete interruption of the axon. So there is loss of conduction. So there is loss of conduction in the new axonotmesis, and in addition to that, uh, axon is crushed. Okay, but here what is intact? Myelin sheath is intact. You see, myelin sheath is intact. Endoneurium, perineurium, and epineurium all are intact, and they are functional. Here you can see Valerian degeneration. Okay, and recovery occurs within some few weeks or few months. Now coming to neurotmesis, means division of the nerve, complete transection of the nerve. It is the most severe form of nerve injury. Here the nerve is completely transected. No recovery is possible unless surgically it is resutured. Okay, and the recovery is also unpredictable. Here the supportive tissue, neural tissue. So all these layers are completely damaged. So according to Seddon, there are three gradings of nerve injury: neuropraxia, axonotmesis, and neurotmesis. In neuropraxia, only there is physiological conduction block. And in axonotmesis, there is conduction block, and in addition to that, axon is damaged. Here you can see Valerian degeneration. And here neurotmesis, there is complete transection of the nerve. Next, coming to the Sunderland classification, which is given in 1951. Okay. So here he has graded the nerve injury into 5 different types of degrees. So what is the first degree? Here there is transient loss of function. Okay, there is physiological loss of function. Okay, it can be reversible within few hours to few weeks since the axon is not destroyed. So it is the first degree is nothing but our neuropraxia of the Seddon classification. Now coming to the second degree nerve injury, there is severe nerve damage, but there is intact endoneural tube. The nerve fiber is severely damaged and is followed by degenerative changes. So the axon is damaged, but there is intact endoneurium. Here, the third degree injury, now axon is damaged and endoneurium is also damaged. In the fourth degree, the axon, endoneurium, okay, and the epineurium all are damaged, and there is disorganization of the nerve fasciculi. Coming to the last one, it is complete transection. So degenerative changes are initiated very early following fifth degree injury to the nerve fibers. So the first degree is nothing but neuropraxia. Second, third, and fourth is they are nothing but axonotmesis. Okay. So it is transient block. It is lesion in the continuity. Fifth degree is the division of the nerve. It is nothing but neurotmesis. See here, the first degree is the most common type of injury to the nerves. It is also called as sudden neuropraxia. It is because of applying pressure over a nerve for a short period. For example, in the Saturday night palsy and a mild form of tourniquet palsy, leading to occlusion of the blood flow and hypoxia. So, if you tie a tourniquet tightly, what happens? Blood flow to that nerve is affected and there is hypoxia. Okay, but in this the axon is not destroyed. It is not, and there is but mild demyelination may also occur. Axon is okay, but mild demyelination may be present, and it is not a true degeneration. This axon loses the function temporarily, which is called a conduction block, physiological conduction block. Then the function will return within a few weeks or within a few hours. Coming to the second degree, it is also called axonotmesis, which shows Valerian degeneration. Here the endoneurium should be intact. So this is the most important point. The endoneurium should be intact. Okay, for the Valerian degeneration. The repair and restoration of the function may take up to 18 months, that is up to 1.5 years. Second degree, this is because of prolonged severe pressure. Okay, prolonged severe pressure. Third degree, here the endoneurium is interrupted, epineurium and perineurium are intact. After degeneration, the recovery is slow and poor and may be incomplete. Okay. Third, fourth, fifth degrees are injury are called neurotmesis. Fourth degree. This type of injury is more severe. Epineurium, perineurium are also interrupted. Fasciculi of nerve fibers are interrupted and disorganized. Regeneration is incomplete and poor. Fifth degree. There is complete transection of the nerve trunk. Recovery is not possible without an surgical interruption, surgical treatment. Now coming to the injury to the nerve cell body. Severe damage of the nerve cell body will result in the degeneration of the entire neuron. Okay, because the proteins are synthesized in the nerve cell body. Because there is no protein synthesis, so no synthesis of cell organelle, the nerve cell will die. In the central nervous system, what happens? The tissue macrophages. Those are microglial cells. They will remove the debris. Okay, and the neighboring astrocytes will replace the neuron with the scar tissue. Whereas in the peripheral nervous system, the tissue macrophages remove the debris and the local fibroblasts will replace the neuron with the scar tissue. So, in the CNS, microglia will remove the debris. The PNS, tissue macrophages will remove the debris. And in the CNS, astrocytes will form the scar tissue. In the PNS, the local fibroblasts will form the scar tissue. Now coming to the stage of degeneration. So the degenerative changes which occur in the part of the axon which is distal to the site of injury. It is called anterograde degeneration or a Wallerian degeneration. It is named after the discoverer Augustus Volney Waller in 1850. He is a neurophysiologist. The degenerative changes which are occurring in the neuron proximal to the site of injury, it is called retrograde degeneration. Okay. These changes will take place in the cell body and axon proximal to the injury. So we will discuss about Wallerian degeneration. Wallerian degeneration is the innate immune response to the traumatic nerve injury. So it is a immune response, innate immune response. So traumatic injury to the nerves in the peripheral nervous system results in the loss of neural functions. So mainly these degeneration and regeneration processes occur in the peripheral nervous system, not in the central nervous system. Repair is achieved through regeneration of the severed axons and re-innervation of the target tissues. The successful functional recovery depends on the ensemble of the cellular and molecular events that develop distal to site of lesion all the way towards the denervated target tissues. Those represent the PNS response to traumatic nerve injury and are termed collectively as Wallerian degeneration after the scientists. So, what are the events which are taking place in the axon which is distal to the site of injury? So, all these events will be included in the Wallerian degeneration. So, what are the changes in the part of the axon distal to the injury? So, it is also called Wallerian degeneration or anterograde degeneration. The degenerative changes will start within a few hours of the after injury. And these changes will take place in the entire length of the axon. Okay, simultaneously it will take place. As we know that they will start within few hours of the injury and they may continue up to 3 months. And they will include the following. First coming to the axis cylinder. The axis cylinder becomes swollen and becomes irregular in shape within a few hours of the injury. After a few days, it breaks up into small fragments. The neurofibrils within it break down into granular debris. So what happens to axis cylinder, that is axon? It will swell and become irregular in shape. Okay, and the neurofibrils which are present in the axis cylinder They will be broken down and they will form debris. Then coming to the next part, that is myelin sheath. It will show disintegration from the 8th day, and the disintegration will be continued up to 32nd to 35th day. This myelin sheath is converted into fat droplets which are containing cholesterol esters. Now coming to the neurilemmal sheath. It is usually unaffected, but the Schwann cells start multiplying rapidly. So neurilemma is intact and it is not affected, but the Schwann cells, they will start multiplying. Now macrophages will invade the region and remove the degenerating axons and myelin and cellular debris, and thus the neurilemmal tube, it becomes empty. Schwann cell cytoplasm proliferates rapidly and it fills up the empty neurilemmal tube. These cells produce a large series of membranes that help to form numerous tubes which play a vital role in the regeneration of the nerve fibers. See this? Here there is injury. Okay. So the axis cylinder swollen and becomes irregular in shape. Now what happens to the fibrils? They are broken. So it is fragmented axon. Then this myelin sheath will be converted into fatted droplets. Endoneurium is intact. Okay. But Schwann cells are there, no? They will start proliferating. And they will send many sheaths of many sheaths towards this side. They will form many sheaths which are helpful in the regeneration processes. See this Wallerian degeneration. This is the nerve fiber immediately after transection. This is the degeneration of the myelin and axis cylinder. So actually there is no complete transection. Okay. Only axonotmesis, lesion in the continuity. Okay. Lesion in the continuity will be there. Okay. Axis cylinder is damaged. That's all. Endoneurium is intact actually. Okay. Don't draw this diagram. Next, what happens? We know that changes in the axis cylinder, changes in the neurofibrils, changes in the myelin sheath, changes in the Schwann cells. Okay. But there is intact endoneurial tube. Now, what happens? Our circulating macrophages, okay, which are present in the blood, they will come into the axis cylinder and they will eat up the all debris which is present in the axis cylinder. So the tube is now completely become empty. Now what happens? The axon, this is the axon. From here it is sprouting towards the damaged part. Now this sprouts of the axon, there are many sprouts of axon which are coming from the normal axis cylinder. So this axon should sprout into the endoneural tube, and the sprouting and entering of the fibrils of the axon, it will be guided by the our sheaths of the Schwann cells, sheaths which are formed by Schwann cells. Then what happens? Myelination of the axon sprout during the process of regeneration. Then all is formed. Then again myelination will be happened. See the same here. Degenerative changes in the distal part of the nerve fiber after injury from A to E. Then these are the regenerative changes. This is the site of injury. Okay. So like this. The previous diagram only it is clear because here macrophages are not shown. And the labeling also not given. See here, this is the intact one. Okay, intact myelinating Schwann cell which is enwrapping the entire axon. See here, these blue colored one are fibroblasts. These are fibroblasts. Okay, this is intact axon, green colored one. Okay, this is myelin, galactin 3 with myelin. So myelin is expressing galactin 3. Okay. Next, what happens? This is the site of injury. Then 24 hours after injury, what happens? Traumatic injury produces immediate tissue damage at the lesion site. Immediately tissue get damaged. Okay. Then what happens? Our macrophages from the blood, they will come. They are galactin-3 MAC2 plus macrophages especially. So galactin-3, formally it is called MAC2 macrophages. They will come from the blood and they will accumulate here within 24 hours of injury. These starved ones, galactin-3 macrophages. Because our myelin sheath is expressing galactin-3, they are attracted. Then what happens after that? Between 36 to 48 hours after injury, destruction of axon is detected during normal Wallerian degeneration. 36 hours after injury. So this is lost. So macrophages eating the debris. So it is becoming empty. The axonal cylinder is empty now. Next, what is this? Recruitment of more and more galactin-3 MAC2 macrophages. Okay, occurs. Myelin disintegration occurs. And galactin-3 MAC2 expression by Schwann cells also begins 48 to 72 hours after injury during normal Wallerian degeneration. So to attract these macrophages, our Schwann cells are also expressing galactin-3 so that they can be attracted towards that. Next, 3 to 7 days after injury, what happens? Galactin-3 or MAC2 macrophages and Schwann cells scavenge the degenerated myelin during normal Wallerian degeneration. And Schwann cells further proliferate and form Bunger bands. I told you, no, the Schwann cells will be proliferated and they will form sheaths inside the axis cylinder to guide the sprouts of the axon which are coming from the proximal part of the injury. So these bands which are formed, these sheaths or bands or membranes which are formed by Schwann cells are called as Bunger bands. So this is the timeline. So within 24 hours of injury, okay, or approximately after 24 hours of injury, macrophages came. And between 36 to 48 hours of injury. So within 1 day, macrophages will come. Then for 2 to 3 days, okay, 3 to 4 days, the macrophages will eat up everything. Then up to 72 hours, what happens? Schwann cells will exhibit the galactin-3. Then more and more macrophages are recruited and they are totally removing the debris. And our Schwann cells will form Bunger bands after 1 week of injury. So what are the cells and molecules which are involved in Wallerian degeneration in peripheral nerve? See this is uninjured now within 48 hours of injury after 3 days of injury. So what happens here? This is dedifferentiated Schwann cell. So after injury within 48 hours, our Schwann cell will be dedifferentiated and it will express some factors, dams, okay, and through toll-like receptors, it will increase the expression of interleukin 1 beta and increase the expression of TNF alpha, and they will cause increase in the CCL2 and MMP9 proteins, and also causes interleukin 6 to be expressed, and it will also increase CCL2. Here it is activated fibroblasts. They will release interleukin 6 and GM-CSF factors. Okay, these are activated by interleukin 1 beta, which is produced from Schwann cells, and they will release galactin 3, okay, which will attract the macrophages. Then after 3 days of injury, what is happening? There are two types of macrophages. The first type will be releasing INOs, okay, and interleukin 1 beta and tumor necrosis factor alpha. Okay, these are pro-inflammatory cytokines. And later on, M2 type of macrophages will be accumulating. They will release interleukin 10, which is anti-inflammatory cytokine. You can see here, within few minutes of axonal injury, initially calcium enters the proximal and distal ends of the nerve and it initiates the axonal breakdown. The axonal breakdown will result in the DAMPs formation, that is damaged associated molecular patterns. They will stimulate the de-differentiation of Schwann cells through the upregulation of JUN and ERB2. So calcium is entering and it is initiating axonal breakdown. These breakdown products will stimulate the Schwann cells through the upregulation of JUN and ERB2. Then through TLR signaling, toll-like receptor signaling, the de-differentiated Schwann cells upregulate the pro-inflammatory molecules. So what are those? Interleukin 1 beta and TNF alpha. They will upregulated. And upregulated activity leads to upregulation of MMP9 and CCL2. The Schwann cells also respond to interleukin 6, which are released from the fibroblasts, which through GP130 signaling triggers an autocrine cascade of LIF and CCL2 upregulation. The fibroblasts respond within hours by upregulating interleukin 6 and GM-CSF, colony stimulating factor, granulocyte monocyte. The later of which this is leads to Gal3 expression by macrophages and Schwann cells. The neutrophils infiltrate the injury site briefly within first day of injury. The endogenous antibodies respond to nerve injury by activating the complement. 3 days post-injury, macrophages will begin to accumulate in the nerve. Activated macrophages will play a dual role in the Wallerian degeneration, which is evident by the factors they secrete. I told you, two types of macrophages will be appearing. Initially, the macrophages will secrete TNF alpha and interleukin 1 beta. They are pro-inflammatory cytokines. CR3 on macrophages binds the complement on a degenerating myelin and it will induce phagocytosis. M1 markers has shown between 1 to 3rd day of injury, M1 macrophages will be appearing. They will secrete interleukin 1 beta and TNF alpha. Then after that, 3rd day, from 3rd day up to 14th day, there are M2 type of macrophages which secrete interleukin 10, okay, which is a anti-inflammatory cytokine. So this will down-regulate the pro-inflammatory cytokines once myelin degradation is completed. So here he is telling the sequence of events. First, what is happening, then what is causing, what is releasing what type of cytokines, and which is activating which one. Okay, so it is more important. So first point is calcium is entering. Calcium is entering the proximal and distal nerve ends. Then it will disintegrate the axon. Then it will release the DAMPs, that is damage associated molecular patterns. These DAMPs will stimulate the de-differentiation of the Schwann cells and they will upregulate the JUN and DRB2. Then through TLR signaling, these Schwann cells will upregulate the pro-inflammatory molecules. What are those? TNF alpha and interleukin 1 beta. They will lead to upregulation of MMP9 and CCL2. The Schwann cells they will also be activated from interleukin 6, which is released from the fibroblasts through GP130 signaling. Okay, they will trigger the autocrine cascade of LIF and CCL2 upregulation. So this LIF and CCL2, all these are pro-inflammatory. Fibroblasts, they will release interleukin 6 and GM-CSF. GM-CSF will leads to expression of Gal3 on the macrophages and Schwann cells. Then neutrophils infiltrate, that is macrophages will infiltrate. Neutrophils and macrophages infiltrate after, within the first day of injury. Okay, and endogenous antibodies, they will also respond to nerve injury and they will activate complement. So 3 days post injury, what happens? Macrophages will accumulate. Okay, and M1 with M1 markers, it has shown some type of macrophages, and after third day they have shown some different types of macrophages which is releasing interleukin 10. Okay, this is the sequence of cellular events in the Wallerian degeneration. First, there is discoloration of the axon through any trauma, anything. And then calcium dependent axonal degeneration occurs with the granular disintegration of the cytoskeleton. Then there will be loss of action potentials. Okay. And retraction of Schwann cell cytoplasm from myelin sheath occurs. Then downregulation of myelin mRNA synthesis by Schwann cells and formation of myelin ovoids. Myelin will be forming ovoid. It will form fat droplets. Then what happens? After 24 hours to 96 hours, there is intense Schwann cell proliferation and recruitments of hematogenous phagocytes, that macrophages and neutrophils. And opsonization of the degenerating myelin occurs by activated complement system without formation of MAC or membrane attack complex. So there is no formation of MAC, but there is opsonization of the myelin by activated complement system. Then removal of the degenerated myelin occurs by hematogenous or resident macrophages and Schwann cells. Then after 10 to 20 days, there is formation of Bunger bands and degeneration of nerves occurs. This is the cytokine network in Wallerian degeneration. That is, this is the time course of the cytokine protein secretion in the normal Wallerian degeneration. You see here, days after injury, this is the percentage of maximum value. So, what is this? This is interleukin 10, which is low initially. Then it is increasing. That is the anti-inflammatory cytokine. Okay. So, it is increased very lately. Then this one is interleukin 6, blue colored one. Then this is GM-CSF. GM-CSF. Then this round one is interleukin 1 beta and TNF alpha. So this both interleukin 1 beta and tumor necrosis factor alpha, they are immediately rising within 1 to 2 days after injury, then immediately falling between 2 to 4 days after injury. After they have fallen, then this GM-CSF and interleukin 6 are secreted in between. After that, last one secreted is interleukin 10. Initially it was low, then it will be increased within 1 week to 10 days. So that is about Wallerian degeneration. Now we have to see the changes which are occurring in the cell body of the neuron. The changes in the cell body of the injured neuron, it will start within 48 hours and continue up to 15 to 20 days. What are those changes? The nistal substances undergo disintegration and dissolution called chromatolysis. This chromatolysis starts around the nucleus and spreads to the periphery. Then Golgi apparatus, mitochondria, neurofibrils are fragmented and disappeared. The cell body draws more fluid, enlarges and becomes spherical. So the nucleus is displaced to one side, that is a periphery. That is, it is displaced towards one cell membrane side. Sometimes what happens? The nucleus is extruded out of the cell. In that case, the neuron atrophies and finally disappears. See here, this is a normal. This is a normal cell body. So now the cell body has swollen and this nucleus has gone to one side. If regeneration is happened, it will become normal, or otherwise if it is extruded, then the entire neuron will be disappeared. Severity of degenerative changes in the cell body. It is variable. The severity of degenerative changes in the cell body is variable. The changes are more severe when the injury to the axon is near to the cell body, and when the injury is caused by forcible tear rather than a sharp cut. In some cases, chromatolysis ends in cell death followed by degeneration of all its processes. If the cell survives, the changes described above are reversed after a period of time. Now, what are the changes in the axon which is proximal to the injury? The changes which are occurring in the proximal part of the axon, it is called retrograde degeneration. These changes are similar to the changes which are occurring in the distal part of the axon. So these changes will be up to first or second node of Ranvier near to the injury. So okay, here in the Wallerian degeneration, the entire axon will be showing these degenerative changes. But in the retrograde degeneration, the changes will be appearing up to first or second node of Ranvier from the site of injury. Now coming to transneuronal degeneration. It is sometimes observed that changes resulting from axonal injury are not confined to that injured neuron, but it will extend to other neurons also, with which the injured neuron synapses. This phenomenon is called transneuronal degeneration. The degeneration can extend through several synapses. For example, it is demonstrated in the visual pathway. So not only that particular neuron, but all the neurons which are having connections or which are synapsing with that neuron, they will also be undergoing degeneration. That is called a transneuronal degeneration, which is demonstrated in the visual pathway. So see, this is the site of damage, it got degenerated. Okay, and this neuron is also degenerated, and it is also classified into anterograde transneuronal degeneration and retrograde transneuronal degeneration. Now coming to the stage of regeneration. The stage of degeneration is followed by stage of regeneration if the circumstances are favorable. It will start within 4 days after injury but becomes more active after 30 days after injury, and it may take several months to a year for a complete recovery. So what are the factors which are affecting regeneration? Regeneration occurs more rapidly when a nerve is crushed than when it is severed and the cut ends are separated. So, regeneration occurs more rapidly in the crush injuries. Chances of regeneration of a cut nerve are considerably increased if the two cut ends are near to each other. That is, the gap should not be more than 3 mm. And there should not be any scar tissue between those two cut ends. So, regeneration occurs more rapidly in a crushed ones, but the gap should not be more than 3 mm and the cut ends should be nearer to each other, and there should not be any deviation of the nerve fiber of the axon, and there should not be any scar tissue between them. Chances of regeneration are more if the two cut ends remain in the same line and are not moved away from each other. Presence of neurilemma is a must for regeneration to occur. Therefore, axons in the CNS, once degenerated, never regenerate as these nerve fibers have no neurilemma. The presence of nucleus in the cell body is also a must for regeneration to occur. If it is extruded, the neuron is atrophied and regeneration cannot occur. Regenerative changes, anatomical regeneration. So, what are the anatomical changes? Changes in the axon. What is the changes in the axon? Stage of fibers formation. Axis cylinder from the proximal cut end of the axon elongates. I said no, the axis cylinder from the proximal cut end of the axon, it will elongate and gives up to fibrils. They may be up to 100 in number. They will extend in all directions. These branches will grow into the connective tissue at the site of injury in an effort to reach the distal cut end of the nerve fiber. So, the access cylinder which is proximal to the cut end of injury will send more than 100 fibrils, and these will go in all directions. Now, this fibril should enter into the endoneurial tube. How it will enter? The strands of Schwann cells from the distal cut end of the axon guide the regenerating fibrils to enter their axon endoneural tube. So, our Schwann cells are forming Bunger bands. It will guide the fibrils of the fibrils which are coming from the proximal end of the axon to enter into the our axon cylinder within the endoneural tube. And once the fibril enters the endoneural tube, it grows rapidly within it. The rate of growth is initially low, that is 0.25 mm per day. Then it increases rapidly once the regenerating axon enters the endoneural tube. That is, it will grow up to 3 to 4 mm per day. The axonal fibers that fail to enter any of the tubes, they will be degenerated. Okay. Then 1 to 5 fibrils will enter the same tube. Under such circumstances, what happens? The largest fibril will survive and the rest fibrils will be degenerated. So, more than 100 fibrils are coming from the proximal end of the axon. Then what happens? Up to 25 fibrils, they are entering into the tube and remaining will degenerate. In these 25 also, the largest one will be surviving and the rest will be degenerated. Stage of active growth. The axonal fibril growing through the endoneural tube enlarges and establishes contact with the appropriate peripheral end organ. The new axon formed in this way is devoid of myelin sheath initially. The process of regeneration up to this stage will take up to 3 months. Then coming to the stage of myelination. This is formed by Schwann cells slowly. This may take up to 1 year. Thus, there is a progressive increase in the thickness of the nerve fiber. But the degenerated nerve fiber, however, it cannot obtain 100% of its original diameter. It can obtain only up to 80% of the original diameter. The axonal regeneration is promoted by growth promoting factors which are secreted by Schwann cells and also adhesion molecules of immunoglobulin family. Okay. In the CNS, the neurons do not require a growth promoting factor for regeneration. Events such as astrocyte proliferation, microglial activation, scar formation, inflammatory reaction following CNS injury are unfavorable for degeneration. Okay. So here in the CNS, there is no role of growth promoting factor because there is activation of the microglia, activation of the astrocytes. Okay. And there will be formation of scar and there will be an inflammatory reaction after the nerves involved if they are damaged in the central nervous system. So all these are unfavorable for the regeneration to be possible in the CNS. So there is no degeneration and there is no regeneration. Here we have Schwann cells. So they will produce growth promoting factors in the peripheral nervous system. Changes in the cell body of the neuron. So up to this we have discussed what are the changes in the axon. Now we came to changes in the cell body of the neuron. What happens? The Nissl granules are appearing again and then Golgi apparatus will also appear in the cell body. The cell loses the excess fluid and regains its normal size and the nucleus again takes its central position. The above changes in some neurons will start within 20 days of injury and will be completed in 80 days. Regenerative changes in the cell body may occur even when the axon does not regenerate. So this is the diagram. First Nissl granules and all cell organelles will disappear and the cell will take the water and nucleus is coming to the one side. Then what happens if it is under the following if it is in regeneration position, then what happens? All the cell organelles will be appearing and nacelle granules will also appear, and our nucleus will be taking its central position. That is anatomical regeneration. Now coming to the functional regeneration. The above described regenerative changes constitute the anatomical regeneration. The function, that is physiological recovery, however, it takes a longer period. What are the complications following nerve injury? There may be complete atrophy of the nerve fiber occurs in severe injuries. And there may be functional complications in the form of misinterpretation of sensations occur when the fibrils do not enter the appropriate tubes, and thus there are faulty connections. So first one may be complete atrophy of the nerve fiber. Second one may be functional complications, that is they are not innervating the appropriate organ. There may be establishment of faulty connections. Next, it may be formation of neuroma. Neuroma formation occurs if during degeneration the nerve fibrils growing from the proximal cut end of the axon do not find their way if the gap between the two cut ends is too large. Okay, if the gap between the two cut ends are too large, or this may be deviation of the cut ends, then what happens? These nerve fibrils cannot reach the distal end. And if the Schwann cells are also not forming the Bunger bands, then what happens? They cannot find the way. Then what happens? These fibrils will turn around in a search of endoneural tube, and they will form a horror-like mass. That mass is called neuroma, and this is a quite painful condition. Now coming to phantom limb. What is phantom limb? It is a feeling sometimes perceived by the patient following amputation of the limb. So if a person got his limb amputation, but he is feeling that the pain is coming from the limb, although the limb is not present, that is phantom limb. So neuroma developed at the site of amputation, when excited, it will cause the patient to feel like the lost limb is present, a feeling of phantom limb. So because of the neuroma, he is experiencing, the patient is experiencing lost limb is present. Okay, that is called phantom limb. Regeneration of the axons in peripheral nerves. So what are the differences between the regeneration of the axons in the CNS and PNS? So in the peripheral nervous system, it is depending on the Schwann cells and endoneural tubes. Okay, so neurolemma and Schwann cells are present. The following mechanisms are involved. The axons are attracted by chemotropic factors which are secreted by the Schwann cells in the distal stump. Growth stimulating factors are also stimulated by released by Schwann cells, okay, which are present in the distal stump. And inhibitory factors which are present in the perineurium to inhibit the axons from leaving the nerve. So these three are important because we have Schwann cells. They will guide the neural fibrils which is coming from the proximal end. They will guide those ones to come into the distal axon, and endoneurium is present. So no fibril will go out of the endoneural tube. Okay, and Schwann cells are present and they will stimulate the growth factors. Now growth factors. Okay, and some inhibitory factors are present in the perineurium so that the axons, they will not leave the particular nerve. Now coming to the regeneration of the axons in the central nervous system. The central axons are not good for regeneration as the peripheral axons if you compare. Why? The regeneration process is aborted by what mechanisms? That is failure of oligodendrocytes to serve in the same manner as Schwann cells. So our oligodendrocytes won't secrete any growth promoting factors and it won't form Bunger bands. And laying down of scar tissue by active astrocytes. So our astrocytes which are present in the CNS, they will immediately form the scar tissue. And there are no nerve growth factors in the CNS. Neuroglial cells, they will produce nerve growth inhibiting factors. Okay, so neuroglia which are present, they will produce nerve growth inhibiting factors. So regeneration of the axon in the CNS is not possible. So up to now we have discussed uh, the degeneration and regeneration in the central nervous system and peripheral nervous system. So the most important one is the Mullerian degeneration. So what are the steps we have discussed? So what are the changes in the distal part of the injury, that is called Mullerian degeneration, and what are the changes in the proximal part of the injury, and what are the changes in the cell body. Okay. So what are the degenerative changes and what are the regenerative changes in the all these parts, and what is the role of Schwann cells, and uh, what is the role of macrophages. Hmm? All this we have seen. The next video is about NMJ, that is neuromuscular junction.